Hi, everyone. My name is Sunny Jain, CEO and founder of Floré Clinical. Very excited to present to you today the microbiome in health and wellness and walk you through some of the key components and findings related to the microbiome and how microbiome analysis and gut health analysis inside of your clinic can be very useful for your patient population. I have with me, joining me today, Dr. Aubrey Anderson, our scientific liaison at Floré Clinical. She has a wealth of knowledge in reviewing microbiome reports, tens of thousands of microbiome reports. Our database at Floré Clinical houses over 40,000 metagenomic profiles, all at the fingertips of Dr. Aubrey Anderson to assist with report interpretation, and she's going to walk you through some of the finer parts of the reporting. Today, we're going to walk you through not only microbiome interpretation and test results, understanding the components of our flagship product, GI Serenity, short-chain fatty acids, the gas producers, the pathogens, but we're also going to share more on how we create that really strong patient engagement through our eHealth platform. And then specific case studies where GI Serenity has been helpful both to the clinician and to the patient, and how we complete that loop in manufacturing custom microbiome modulating solutions, your polyphenols, your resistant starches, your oligosaccharides, and putting those together in a meaningful way to drive a beneficial shift of your gut microbiota. Now, your body is composed of trillions of human cells, and every one of those cells contains its own DNA. But from a human cellular perspective, each of those cells and the human DNA contained within them is identical. Over 30 trillion human cells have identical DNA across the cells. That's what makes you, you, and all the biology and footprint of that information. Now, interestingly, we've found that your body is composed of trillions of microbial cells. Some used to say it was 1 to 10. It's now well understood that this is more 1 to 1. So an equal number of cellular representation of your body are actually microbial cells, 30 trillion microbial cells. What's so interesting about those microbial cells is that they house DNA, but those microbial cells don't house all the same microbial DNA elements. They're actually really diverse across the cellular population. And between two individuals, you may only be 10% unique. You're 90% different from your microbial DNA of your gut microbiome. Now, as we began to understand more and more of this science and the importance of the gut microbiome, scientists and leading clinicians began to publish in an impactful way about how this is changing human health. In 2016, when we founded Floré Clinical, we knew that you could modulate the gut microbiome. And we understood that metagenomics was a key part of bringing this type of biomarker testing to commercial market. And so we started on that path and spent about 4 to 5 years in research and development before launching GI Serenity. And what we've seen in that time is an exponential ramp up in the peer-reviewed literature around human gut microbiome impact and its clinical utility, topping off at over 20,000 publications every year for the last couple of years. That's an astounding level of knowledge and understanding of the gut microbiome. Now, what we do at Floré Clinical is actually mine all of that information through a natural language processing algorithm that we've developed. And we can take that peer-reviewed literature, which could be potentially impossible to review 20,000 peer-reviewed literature in 1 year, and extract all the text and information and tabularize that and make that simple for our database to be able to understand the most impactful areas that are coming out related to microbiome science. And what we found is that from birth all the way through later in life, there are aspects to our wellness that are correlated, associated, or even directly tied to the microbiota that are living inside of our intestinal tracts. And we see this not only in gastrointestinal issues which is expected, but we see these in areas as broad as the gut skin and acne and the gut allergies, immune system connection. We're finding more and more science related to obesity and metabolic health, the gut-brain axis with depression and autism. We're seeing so many more new indications even come out recently, indications related to Alzheimer's, Parkinson's, diabetes management, and of course, general IBS and IBD. So it'd be really important as we age to continue to monitor and understand how our microbiota shifts over time as we go from a child into adult microbiota, and as we shift from an adult microbiota later in life as we age every decade. That microbiota is so unique to you. And so your unique microbiota is shifting and changing over your lifetime. And so monitoring of that unique self becomes critically important in managing one's wellness. Now, there's different ways to monitor the microbiome. And let's define a few terminology before we get too deep into the science here today. The microbiota is a term used to describe all the living cells of these microorganisms that are living inside of the gut intestinal tract. The microbiome is considered the DNA elements of all of those cells extracted out and looked at and analyzed as it represents that microbiota. This is why we find DNA analysis critically important in order to understand what's going on in the microbiome. Now, there are always ways to look at biomarkers, whether it be microbiome or whether it be human analysis. And this is the central dogma that we're looking at here. We have DNA, and our cells have DNA, and they transcribe. And the transcription of off of the DNA represents expression, which is RNA, and you can measure RNA, sometimes called whole transcriptomics in microbiome analysis. But ultimately, we're trying to understand the metabolites or protein that gets translated off of those RNA molecules to understand how these protein molecules or these metabolites are interacting with our human physiology and changing our physiologies. Now, we don't always measure information in testing with protein. You may have heard of things like ELISA assays that are measuring protein. You may have heard of RNA assays that are single gene expression assays that measure things like cancer gene representation. But we measure DNA, the most stable of these molecules and the most easily to be able to detect, because it's isolatable, it's reproducible from an analysis perspective, and it has the robust clinical criteria to constitute acceptable clinical biomarker for future use. Now, it would be wonderful to analyze all of the human transcripts of our body at all times to be able to understand it. Unfortunately, the technology is just not there. It's not currently utilized in a broadly adopted sense of people measuring their own human RNA and their entire human RNA transcriptomics. We think it's wonderful research. It's got 10 to 15 years to go. We're excited for this field and we hope it continues to advance. Metabolomics, or the protein types of analysis, falling into a similar type of boat. Trying to analyze all of these metabolites all at the same time can be very difficult to achieve clinical sensitivity and specificity. And as that technology continues to evolve, we'll begin to see more progression in protein analysis. We're very excited for what the future has for transcriptomics and metabolomics, but let's start where we know quite a bit. In all of those publications that I was referring to previously, majority of those are isolating and looking at the DNA of the microbiota. So our understanding and clinical utility really comes from the analysis of DNA and leveraging that into clinical studies and clinical meaningful reporting. Taking a look at our history of how we got this exciting test to market, in 1977, we used to be able to do things like 16S and 18S analysis These are small little regions of the bacterial genome that you can design PCR primers for, amplify off of those regions, and be able to determine the taxonomy, the identity of that microbe or which organism it is. Now, it was generally done one by one at that time, and we've had evolving technology that allows us to now do this in a broader parallel sense. In PCR innovation, we're able to identify targeted microbes very quickly. And then by 1992, qPCR began to become a commercialized type of tool where we're starting to look at fluorescence coming off of the amplification and using that laser technology or light technology to read off of that fluorescence and understand down to very sensitive levels of detection what elements of DNA or RNA are present. But again, amplifying off of a genome structure. And every time we amplify, we're going to begin to create a distortion, similar to a mic and a speaker. In between, you have an amplifier to help propel that, that audio through that microphone. But in that propelling of that audio, you create a distortion of what comes out of the end speaker. Now, we've made speakers as good as we can make them, but and we've made PCR as good as we can make it. But there is that distortion that always exists, and it essentially creates bias. These primers are coming down, binding to the genome, and then amplifying select regions where the primers have the best homology and just selectively amplifying that region. And that may not be the specificity that you're looking for, and it may be variable between time points and tests. And hence the advent of whole genome sequencing. Whole genome sequencing looks at the entire genome of these bacterial organisms, not just these short segments that are defining their unique taxa, but actually sequencing through the genes, the promoters, the antibiotic resistance cassettes, looking at these various regions so you can understand the genetic function of these organisms and get more information and more sensitivity and specificity to the species level resolution. You can kind of think of PCR and qPCR and these 16S types of technologies as the way we used to watch television with the rabbit ear TVs. Wonderful technology, definitely an advancement from black and white to making it to color. But now we have 8K OLED technology, and it's very affordable and cost effective to have one in almost every room in the house. And that's why we believe in whole genome sequencing for analysis of the microbiome. As the cost of sequencing has precipitously dropped, it's allowed us to analyze the microbiome in this high resolution way. So now that we've established the different types of biomarker testing that make microbiome analysis clinically important, how do you actually determine which solution to utilize and why you should consider custom solutions over the one size fits all? Some of the conditions that we've mapped early on from the scientific literature, but also now mapped in our own proprietary database that individuals and patients are utilizing gut microbiome testing for include chronic gut issues. So these could be things where you see cyclical occurrence of inflammatory microbes continuing to come back. There are patients that have reported through our health and diet survey of having chronic pain. We've had patients that report having skin health issues. ASD is a study that we've done in collaboration with Arizona State University and published in M Systems in 2024. And then we've seen interesting areas in the gut-brain axis, in mood, as well as in metabolic health with obesity and diabetes. All of these areas are continuing to mature, but just help us understand why are patients choosing microbiome analysis and why are clinicians choosing microbiome analysis to continue to explore holistic solutions for their patient base. Now, one of the areas in metabolic health and obesity that we're seeing increase over time, the rate of obesity in the adult population and as well as in the children population, over 40% in adults, over 60% in children. The root of that, as identified by the scientific literature, could be found in the consumption of ultra-processed foods, or what some are calling the standard American diet or the SAD diet. These ultra-processed foods have a way to feed the organisms of the gut in a negative way, or the organisms that are not beneficial for our human health, and repress the organisms that are commensal, that are beneficial for our health. These specific metabolites that the beneficial organisms release actually are able to contribute to our human health. As much as 30% of metabolites that are released by the gut microbiota can be found in our circulation, setting the stage for understanding the microbiota could help us understand some of these mechanisms in obesity and metabolic health. Now, when we began to dive into some of that mechanism, what is actually happening? How do we look at the biochemistry of these complex carbohydrates or other molecules that could stimulate healthy bacteria, or vice versa, inhibit some of the harmful bacteria? When looking at the healthy bacteria, we found these three very clear categories of fibers that can be beneficial: complex carbohydrates, resistant starches, and oligosaccharides. What's interesting about these specific dietary fibers or resistant starches or oligosaccharides is the organism that they will in turn drive up the concentration of in your gut microbiota is very specific. You can take a broccoli powder and see some general benefit, but what it's actually doing in the gut microbiota is it's driving up the concentration of these butyrate producers in the gut, the Faecalibacterium prausnitzii, the Ruminococcus and Lactobacillus organisms, and the species contained within. And as we began to provide synbiotics, the prebiotics and probiotics combined, we were starting to see these shifts in longitudinal data analysis that we've done. And that mirrors very closely to the healthy diets that nutritionists and doctors have been recommending to us, where we need to implement more of these nutrient-dense healthy greens, these legumes and other whole grains into our diet, perhaps thinking that they were feeding our human cells, but what has in turn, in part, been found to be feeding the other half of our cellular mass, our microbial self. And the same goes for resistant starches. You have potato starches, green banana powder, other grains that will feed specific gut microbiota like Rosebaria or Akkermansia. Some of these organisms have strong associations to GLP-1 like agonists and receptor agonists, and others are also producing short-chain fatty acids like butyrate, propionate, and acetate that are stimulating that mechanism and enabling healthier outcomes for metabolic health. Another specific way to modulate the gut microbiome, and a long-standing way that people have defined as prebiotics, are oligosaccharides. So there's fructooligosaccharides, galactooligosaccharides, human mammal oligosaccharides. There's all these different types of long branch chain oligosaccharides that are difficult to quickly dissolve and break down, allowing the bacteria to use them as a fermentation source, essentially increasing the concentration of these organisms. Generally, they're going to increase the concentration of bifidobacterium and these immune stimulating microbes of the gut. And if you can increase the bifidobacterium and immune stimulation, you could solve for some of those underlying issues that have immune regulation involved in them. And there are other ways to do that through diet as well. But using these types of tools in combination and knowing which ones to put together at the right moment for what the microbiota is telling you is what we're able to assist with at Floré Clinical. Now, when you look at this mechanistically, we're finding that that the low fiber diet in the literature has strong associations to creation of aberrant metabolites, essentially metabolites that are not helpful for your human body. And if you can have high levels of commensal organisms, your Roseburia, your Faecalibacterium prausnitzii, some of these other bifidobacterium organisms, and perhaps even some eubacterium as well, you can create an environment where there are metabolites called short-chain fatty acids that are benefiting the intestinal lining and the tight junction intestinal epithelium. And these short-chain fatty acids not only are able to provide interintestinal benefit in feeding the right metabolites that act as prebiotics for other organisms, but also have direct binding sites to the intestinal epithelium. And this is research that was done as recently as January of 2024, where scientists have actually now shown that the biochemistry of these short-chain fatty acids, specifically butyrate, have binding structures inside these GLP-1-like receptors. So this is fascinating research coming out, and it mirrors very closely to some of our findings analyzing our database as well. So if these short-chain fatty acids are critically important to be a part of the molecules inside of our intestinal tract, we need to start to consider what are the ways that we can naturally stimulate the production of this short-chain fatty acid instead of end-arounding that whole mechanism through direct stimulation of GLP-1. Perhaps there's a natural way to build up the homeostasis or the specific organisms of the gut that are going to produce all these wonderful short-chain fatty acids, ultimately leading to the metabolic benefits. That molecule butyrate has now been implicated as a central conduit in various different types of organs, not only coming from the intestinal gut, but then triggering downstream pathways across that gut barrier through the lamina propria into the tissues and eventually getting into the circulation and other organs of our body. That downstream pathway has now been shown to have positive and negative effects. And this just shows how central it is to areas that are critically important in obesity management and GLP-1 receptor pathways, not only affecting the liver, pancreas, adipose tissue, and the brain, but stemming from its source, the microbiota they're producing it inside of the intestinal tract. So when we took our database of over 40,000 metagenomic profiles, looking at the microbiome at deep levels, and then selectively isolating out all of the individuals that have elevated levels of BMI index, establishing an obesity cohort, and comparing them to our healthy cohort, where we peer-reviewed and published on in IBS as well as in autism, and then further trimming that healthy cohort for the obesity study. And what we found is that there is a strong statistical difference in the gut diversity between individuals that represent the quote-unquote healthy cohort, individuals that don't have self-reported chronic condition or management, versus the obesity cohort, these individuals with elevated BMI. This was fascinating to see and begins to establish the point that perhaps the microbiota in the context of obesity is different. The composition of it is different, and the metabolites that it's going to release are aberrant, and we need to move it over to healthy. So after analyzing the healthy cohort and the obesity cohort, we dove further into the microbes that are differentiating between these cohorts. What are the specific organisms? And we started to categorize them. And we found that there are three main categories of these microbes that are different. There are ones that are producing short-chain fatty acids. So there are less short-chain fatty acid producing microbes in obesity. There are more gas producers in obesity. There are more pathogenic microbes, and their subsequent toxin production in obesity-related cohort. So if we can begin to solve for these three axes in obesity paradigm, so this sets the stage for there being three key areas of concern related to the paradigm of obesity treatment and the gut microbiota. Looking at your short chain fatty acid microbe production, looking at your gas producers, looking at your pathogenic microbes could be critically important in a holistic treatment paradigm for patient care when trying to resolve root cause of what's going on related to obesity. So I'm pleased to present GI Serenity, the report that can actually provide insight into these key areas of the microbiota, and what's more, get all the way through into the detail of eukaryotes, archaea, viruses, other bacteria that could be lending to the issues holding back a patient from feeling their best. And we pair this with our eHealth technology, and I'm pleased to introduce Dr. Aubrey Anderson, who will be taking it from here, walking us through some of the finer points of GI Serenity.
I'm pleased to present to you 3 case studies where we utilize the GI Serenity test to identify the 3 genetic signatures that we found were common in patients with obesity, and then go through how you identify this through the report, and then solutions for these patients based on custom probiotics. We have a 72-year-old male. He complains of gas, bloating, brain fog, and fatigue. His health history includes reflux, arthritis, rosacea, and seasonal allergies. In terms of food and sensitivities, we have eggs, dairy, and salicylates. He takes PPIs and statin drugs. He's on a multivitamin, takes calcium, magnesium, vitamin C, vitamin D, and an omega-3 supplement. He's a non-smoker. He drinks 1 drink per day or 7 drinks per week. When we asked about diet, he identified no specific dietary practices. We can assume he's probably on a standard American diet. And in terms of exercise, he quantified himself as moderately active, exercising 2 or 3 times per week. This patient's BMI is 34.86. So he is one of our obesity patients. And so let's run a test on him and see what his gut microbiome shows us. Before we dive into the analysis, let's take a closer look at this patient's diet. They said no specific dietary practices, but when we asked them greater detail, we found that this patient's greatest sources of protein are chicken, beef, fish, and pork. They eat green vegetables every day, several times per day. They also eat saturated fats, and these could be animal proteins, fried food, dairy, coconut oil, pastries. He's eating those several times per day. Unsaturated fats, he's only eating a few times per week. And the fermented probiotic foods he identified in his diet were sauerkraut, yogurt, and soft cheeses. He is taking a probiotic. It's an off-the-shelf probiotic. He has not taken antibiotics or Accutane for greater than 2-week period of time. And he has not taken any antibiotics in the last 3 months. We ran a GI Serenity report on this patient, and the abnormalities that were shown were really high abundances of 2 notorious gas producers, Bacteroides vulgaris and Bacteroides fragilis. And so you can see from this report that his abundance is almost 3 times more than the upper limit of what would be considered normal for Bacteroides vulgaris. And you can see his Bacteroides fragilis is also much higher than average. It's highly likely that these microbes are causing this patient gas and bloating and digestive discomfort. And so now that we've identified the exact strains that are overgrown, we can develop a treatment strategy through probiotics to address this overgrowth. With this patient's test results, our formulation scientists were able to put together a custom probiotic formulation specifically designed to help this patient reduce some of those gas producers by introducing competitive strains that help reduce the numbers of those Bacteroides vulgaris and Bacteroides fragilis. So in the graph that you can see on the right here at time point 1, that is the gas producer's abundance when the test was first taken. After we administer the custom probiotic, you can see that each follow-up test at each time point, the gas producer's abundance changed significantly. And by the time we got to time point 5, which is about a year and a half into treatment, you can see the gas producers have reduced to less than 2. This was very exciting to see how a probiotic can interact with the gut microbiome and actually cause this shift and bring down the level of gas producers for this patient. For our next case study, we're going to look at a patient with obesity that has the microbiome signature of pathogen abundance. This is our patient. He's a 35-year-old male. He's complaining of frequent bowel movements, gas, and brain fog. He suffers from acne, depression, and seasonal allergies. His food and allergies include eggs, dairy, nuts, and beans. He takes no medications, takes no supplements. He has 1 to 3 alcoholic beverages per week. He is practicing an intermittent fasting diet. He is dairy-free and mostly vegetarian. He exercises 2 to 3 times per week, and his BMI is 35.39. When we dive into this patient's diet a little further, and we asked him about some of the content of this diet, we found that he gets most of his protein through plant protein, and he eats meat, fish, and chicken just on the weekends. When asked about eating green vegetables, this patient was honest with us and said almost never. But they do eat saturated fats less than once per day, and unsaturated fats just a few times per week. The probiotic foods that they are including in their diet are kimchi, kombucha, sauerkraut, and yogurt. This patient does take a probiotic. It's one that's off the shelf. They have had prolonged antibiotic therapy or used Accutane for greater than 2 weeks over the course of their lifetime, and they have not taken any antibiotics in the last 3 months. So let's take a look at their test and the abnormalities we found. Here you can see that we have multiple microbes that are abundant that are known problems. And so this Citrobacter you can see is elevated. This is the one in the first row. And you can also see that Klebsiella pneumoniae is also elevated in this patient. Now that we've identified the problematic microbes, you can develop a treatment plan to address them. And you in your practice, depending on, of course, which problematic microbe you may find, you may decide to treat these patients, according to your own treatment. However that is, as all providers, we all treat differently. We don't always have the same exact plan. But one thing most of our treatment plans include when we're working on things like the gut is a probiotic. And so let's pick a probiotic for this patient based on their pathogen abundance. For this patient, we created a custom formulation that is mostly comprised of lactobacillus species and bifidobacterium species, and the prebiotic filler in their formulation was the powerful antioxidant turmeric. Time point 1 is the pathogen abundance when they took their test, and then time point 2 and beyond is the pathogen abundance after the probiotic was administered. And you can see, again, these time points are about 4 to 6 months apart. You can see the significant drop in the pathogen abundance for this patient right away. Even within that first sample point, it was a significant drop. And so through a subsequent follow-up testing, you can continue monitoring these pathogens to make sure that their populations are staying in check. For our third case study, let's take a look at a patient whose GI Serenity report shows low short-chain fatty acid producers. Here is our 47-year-old female patient. She is suffering with heartburn, constipation, frequent diarrhea, fatigue, and brain fog. Her problem list includes migraines, IBS, and recurrent UTIs. She has no known drug allergies. The only medication she takes is zonisamide, 100 milligrams by mouth 3 times a day. In this medication, she's taking it off-label for weight loss. The supplements she's taking are ashwagandha, turmeric, and magnesium. She's a non-smoker, has 1 to 2 drinks per month. For her diet, she disclosed that she has no specific dietary practices, so we can assume it's probably standard American diet. Moderate exercise 2 to 3 times per week. And this patient's BMI is 34.49. So let's take a deeper look into her diet and see what she's eating. For this patient, her main sources of protein are chicken and beef. She eats green vegetables once per day. She eats saturated fats once per day, unsaturated fats a few times per week. She doesn't eat any of the fermented or probiotic foods. She does not take probiotics. She has had prolonged antibiotics use or Accutane use. She is not currently on antibiotics, but did take some within the last 3 months. So in her report, you can see some of our most important short-chain fatty acid producers are extremely low. This means that this patient is missing out on all of the benefits and anti-inflammatory benefits of the short-chain fatty acids. It also means that the gut integrity is potentially in jeopardy because it's not being nourished by these metabolites. And so, but all 3 of these are some of the most important short-chain fatty acid producers, and you can see that this patient is really lacking in these. And so let's take a look at what we did for this patient and how her short-chain fatty acid producers responded. Okay, to bring up the short-chain fatty acid producers in this patient, the custom formulation included psyllium husk powder and turmeric, several species of bifidobacterium, Streptococcus thermophilus, and lactobacillus. Time point 1, you can see that her short-chain fatty acid producers were barely detectable. It's that tiny little sliver of a line at the bottom. And by time point 2, which was about 6 months later, you can see that she had a significant bump in her short-chain fatty acid producers. And then by her 3rd time point, you can see that she had a tremendous turnaround in the growth of those short-chain fatty acid producing microbes. And so now she's getting all of the benefits of those short-chain fatty acids, the anti-inflammatory benefits, and the enterocytes lining her intestines are now getting all of the benefits of those short-chain fatty acids to promote gut integrity. So now that you've identified these problematic microbes for your patients, are you ready to pick a probiotic for your treatment plan? Perhaps you've been here. I know I've been here. We call the wall of confusion, overwhelming number of probiotics off the shelf with all sorts of different strains in there. And I've spent quite a bit of time in my practice trying to match a probiotic for my patient. But it really turns into sort of a probiotic paralysis where it's really hard to make a decision. And that's why I want to introduce the GI Serenity by Floré Clinical, because it gives you a pathway to do this that is much easier. So once you've used the GI Serenity test to identify problematic microbes or microbes who you want to foster growth with, our team of formulation scientists will create a formulation based on your patient's data from their GI Serenity test, as well as the health history and conditions that this patient is dealing with and suffering with. They create this formulation blending those 2 things together. So your patient gets 100% custom probiotic that is to target and treat the issues that they are dealing with based on their most recent report. Once our formulation scientists have created your patient's custom formulation, those 100% custom probiotics are drop shipped directly to your patient's door. The benefits of having precision formulated probiotics is that it removes the limitations of having to stock tons of formulas in your office and then try to play mix and match to find ones that are perfect for your individual patient's unique microbiome. These custom formulations are created by our scientists based on your patient's individual microbiome report and their individual health concerns. So rather than lumping them into a bucket, they're getting 100% focus on their individual microbiome signature. The acid-resistant capsules that these probiotics are housed in will protect these live strains through stomach acid so that they open in their destination of the small intestine intact, and our strains are alive so they can go do work in the places we want them to be. That acid-resistant capsule is incredible for increasing the survivability of these probiotic strains so they will take up residence in your microbiome. All custom formulations are formulated with the patient's allergies, sensitivities, and dietary restrictions in mind so that they get a product that fits for them. The premium ingredients in these probiotics are sourced from industry-leading suppliers such as DSM and Frontera. To ensure the highest quality in these ingredients, all ingredients are stringently third-party tested to make sure that they are acceptable before we use them. We are a UL certified facility that does this third-party testing. This testing also includes checking for things like heavy metals and contamination. We also do our own DNA strain verification to make sure that the strains that are going into the probiotics are exactly what they say they are. Every time your patient does a new GI Serenity test and gets a new report, they will get a brand new custom formulation based on the newest test results so that your patient's custom probiotics are keeping up with their evolving gut microbiome over their lifetime. Our interactive patient portal allows you and your patient to collect clinical insights beyond just a PDF or a printout. This interactive portal allows you to search for a specific microbe. So if there is one that you wanted to look for that maybe didn't show up in the top 10, but you're curious, you can put that microbe into the search bar here, and it will identify it in that patient's sample if it was present. You also can track the abundance of an individual species over time through this portal, so you can monitor how you're doing with them between each time point to assess if your treatment plan is working. You can also use this interactive patient portal to learn more about the individual microbes, how they're classified, some of their functions and the purposes that they serve, and the diseases or conditions they are often associated with. We have an eHealth portal that allows you and your patient to stay engaged with their gut microbiome report and their subsequent testing so that you can keep track of their results and keep track of the ingredients in their newest formulations when they're created. Thank you for joining us today to take time to learn a little bit more about the gut microbiome and the value of whole genome sequencing in that analysis. I hope you also learned a bit about custom probiotic formulations and their value in addressing different microbial issues like the gas producers or pathogen abundance, or identifying the lack of short chain fatty acid producers in your patients. We are looking forward to partnering with you and your patients on their gut health journey.